Thanks very much, Brian, for that introduction. And thank you for the invitation to be able to speak today at the end of these really wonderful talks. I mean, this has been, eye-opening for me to see all the younger people really working at this disease, and the breadth of the research has really been enlightening and invigorating to see. And it's really a hopeful time, I think, for understanding this disease and moving forward. And Richard, I appreciate, I don't know where you went, but the summary of really looking at therapies for these diseases and all the problems associated with it and the multiple targets and how we have to think about different diseases. Those are things I was planning to highlight, and I appreciate that you've sort of set the stage for me. My plan today is really to kind of talk about some of the research that we've been doing to try to understand some of the approaches to therapy with prion disease. And as you saw today, there's really several approaches, and that's going to be my end message for this talk. But what I'll do is lay the foundation for understanding where the targets might be approached in prion disease. So I'll go over, and I know everyone knows this very well. It's been repeated several times, but I'm going to go over the underlying process of prion disease, identify some of the areas that we can target for therapy, and then talk to you about some of our failures and some of our successes. Okay, and this is the campus of the University of Chicago, and this is my office and lab. And you're welcome. Anyone is welcome to come and take a look. I can give you a tour. It's a beautiful campus, and happy to have you come and see the place. Okay, so here's the little review. We can think about prion disease as a two-step process. There's the generation of prions, and then there's the propagation or the spread of prions throughout the brain. So the propagation occurs from, as you well know, a misfolding of the normal prion protein in the brain to an abnormal form. And we call this a misfolded isoform of PRP or PRP scrapie, PRP-SC. This occurs primarily through spontaneous conversion. It can occur in about 15% of cases that are due to a gene defect in the prion protein gene, and rarely through exposure in the environment. Once you form this misfolded isoform of prion protein, that can now have the potential to interact with other normal prion proteins, convert them to the abnormal form, and this eventually accumulates, causes cell death, and leads to the clinical symptoms of prion disease and the pathology and abnormal protein associated with the disease. Now, one thing I want to bring to your attention, I know this is the CJD Foundation, but there's more than just CJD to prion diseases. And it's important. CJD is the primary, or the most common of the prion diseases, but there are several other phenotypes or subtypes of disease that we think about. Gerstmann-Straussler-Schenker disease, or GSS, fatal insomnia, variant CJD, and variably protease-sensitive prionopathy. And so why do I bring this up now? As you can see, there's a pointer here, right? Okay, there we go. Okay, as you can see, there's several different pathologies associated with each of these subtypes. So on the surface, these look like very different diseases. And the clinical features of the disease can also differ among the different subtypes, as can the age at onset and the course of disease. So I want to just bring that to your attention that there are subtypes of disease that present differently and behave differently, and we have to think about those, I think, in developing therapies for these diseases. Some therapies may work for one subtype, but not for the other. Okay, so now this brings it down to a very basic level and identifies the targets that we can maybe approach. So there's the conversion. We can inhibit the conversion. We can delay or inhibit the misfolding or the propagation of the protein and increasing PRP scrapie. We can enhance the protein degradation. And we can inhibit, as we saw today, maybe the cell death pathway and preserve neurons. And so all these are obvious ways that we can approach therapy with prion disease. We tend to look at the beginning of this schematic because the earlier, and that was stated, the earlier we can treat the disease and stop the disease before it gets to the cell death pathway, the better off the patient will be. So we focused on those two areas primarily throughout the years. This is one of the experiments, a study that was done a little while ago, and this looked at a compound called Onlay 138B. Onlay 138B was identified at the University of Munich by Armin Gies in his lab, and he graciously allowed us to use it in this experiment. And I'd like to mention the Cure FFI Foundation supported this work, and I appreciate that. What we wanted to do is see there was potential for this compound to stop the aggregation of prion protein. So it's identified as an oligomer modulator, and this simply breaks up aggregating proteins. So we thought, let's look in a transmissible model and compare it to a genetic model. To answer that question, can a therapy work across subtypes? So there was evidence that it would work well in a transmissible model, and you now know that these models occur by inoculating prions into mice and then monitoring them for the onset of disease and death. And as you can see, compared to mice treated with placebo, animals treated with Onlay 138 lived much longer, almost twice as long. So it extended the life of the animal quite significantly. And at the time period of 140 days, when the animals were sick on placebo, you could see extensive pathology in those animals, but no pathology in those treated with Onlay 138B. So this seemed very promising, and our hope was that this would work in a genetic model also. This is our genetic model of GSS. And so one of the characteristic features of GSS is ataxia of gait, clumsiness in movements. And you can see mice that are normally very agile, this mouse is having lots of trouble ambulating. He's clumsy, he's falling. And this is the earlier stages of disease, and eventually this progresses over time. So we tested, and they also show the pathology that's characteristic of GSS, which are these large amyloid plaques deposited primarily in the cerebellum, which controls our coordinated movement. We tested this in the animals and treated them with Onlay 138 in a similar way that we treated the transmissible mice, but unfortunately there was no change in the onset of disease or the time of death. So the lesson we learned, even though it was unsettling that it wasn't useful in that model, but the lesson we learned is that we need to account for disease subtypes when we think about these therapies. Now, one of the other things that I've been doing in the lab for many years is looking at that second step, the propagation of prions. And so the goal here is to try to understand how these two molecules come together. The bad form of the protein, the misfolded isoform, and the normal form of prion protein. How does this interact? Are there specific sites that are most important for the interaction of the protein? And if there are specific sites, can we identify those and maybe block the interaction and block the propagation of the protein? So we used yeast to study this interaction of these proteins. And yeast is a simple cell model that you can express the protein in the cytosol of the yeast. And when we do that, we can create aggregated prion protein. And you can see that this is prion protein tagged with a fluorescent marker compared to the fluorescent marker alone. Aggregates nicely in the yeast. And it has the properties that are characteristic of the misfolded protein in prion disease, protease resistance and insolubility. So we use this to screen a variety of modifications of the prion protein, and we settled upon some variations that involve deletion of a certain region of the protein in the central region of the protein that did have some evidence for its importance in the protein, but we isolated it down to an even smaller area in this 8 amino acid segment of what is essentially a palindrome of alanines and glycines. When we did that, we found that there was no prion protein misfolded form, aggregated, or protease resistant in either of those yeast. And so we asked, could this be a prion protein that doesn't form prions? Could it propagate prions, or could it block propagation? So we made transgenic mice that express this deleted segment. And the first thing we asked is, do these mice survive okay? Because another mouse model, so this was taking a big chance, another mouse model deleted that larger area of 105 to 125, and it was lethal, and the animals didn't survive. Our animals survived beyond 800 days with no pathology in their brains, so that was a good thing. So the next thing we did is challenge these animals with prions to see if they develop disease. And you can see that after 600 days, these animals were not symptomatic, and there's no pathology in the brain, compared to animals that had normal prion protein expressed at a similar level of these mice, showed lots of pathology and died. So there's more to come on this. I'm not going to talk too much more, but there's lots of experiments that we have planned for these mice in hopefully preventing prion disease and blocking it. So I see that my clock is ticking very quickly. So what I'm going to do is speed it up a little bit. And you can tell my voice, got a little bit of a sore throat here, and my voice is going right now. Okay, so we also looked at protein degradation. How can we harness what the cells normally do to try to get rid of bad proteins? Can we amplify that or enhance it and get rid of prion protein more efficiently? And so, long story short, we enhanced the process of what's called autophagy. And this is a process that the cell does normally and helps to remove misfolded aggregated proteins in the cell. And when we did that and we looked at our GSS spontaneous model, we saw, in fact, that at every time point they had lower disability scores and they had a delay in the onset of disease, significantly 18% delay. And when you consider that these diseases occur in early in life, that could be a considerable change. Maybe instead of 50 getting affected, the age of 60, the disease will start. Still not a cure or a perfect cure, but maybe an enhancing element to other treatments. Okay. And then finally, what most people have focused on here today and talked about really is looking even earlier in this pathway, right? And lowering the amount of PRP that's expressed in the brain, because that's the substrate that gets converted to the abnormal form. So let's get rid of it. But what we focused on is not PRP, normal PRP, non-mutated PRP, but genetic forms of PRP. So our goal is to create a therapy that is like a designer therapy, essentially, for people that carry mutations of the prion protein gene and selectively targets that gene. And so you only have one copy of the mutated gene, as was discussed earlier today. And the idea is to selectively inhibit that copy from being expressed and leaving your normal copy of prion protein gene to express throughout life. And why would that be important? Although there's not evidence for it, we don't have a human model for lacking PRP during their whole life. The importance is that this treatment would be a lifetime treatment. We can identify carriers early in life because they carry the gene mutation, and then we can initiate therapy at an early stage. And this can be, as you've seen, with viral vector delivery, and have the therapy be expressed in the brain for the entire life of an individual. And so rather than just shutting off all PRP genes, both PRP genes, we want to shut off just the mutated gene. And so we're using a process of allele-specific RNA interference that basically prevents the RNA that signals the development of the protein from surviving. And there's an effect in the cell, depending on based on the therapy that we provide, to degrade the RNA so that the protein is never produced. And we made selective therapies or selective sequences that will target only the mutated gene. And here you can see an example of the one that we selected. And this is a cell culture expression of normal prion protein, the RNA, and then the protein. And here we see normal prion protein expressed and normal RNA. But because this is selective for the mutated gene, we can see that in the mutated prion protein that's expressed in cells, the RNA is essentially turned off, as is the expression and the level of the protein. So it seems highly selective. And now the test is to put it into mice. And we use the viral delivery systems that people have talked about earlier, adeno-assisted virus. And this is an older picture of some of the problems that we have found along the way, that the delivery of this therapy is present, but somewhat on the low side. So you can see this is the fluorescent marker that targets where the virus is gone and the therapy is being expressed. And unfortunately, that this low level really didn't affect disease outcome in these animals. But over the years, we've modified our sequence. We've changed the protocol, and there's been new vectors developed that allow us to now have this picture most recently, and you can see the intensity is much greater. And in fact, we're very happy to see, and this is really hot off the press, that we now have a reduction of from 40 to 50% of our mutated prion protein expressed in the brains of these animals. And we're now waiting to see whether this affects the disease itself. So we're hoping that this is going to significantly delay onset and symptoms of disease. So rather than summarize all those, because they were really summaries of experiments, I just wanted to give you the take-home message that there are really many options and possibilities to change the course of prion disease. And I'm even more hopeful after hearing the talks today that this will get done. And just to thank you to my deceased puppy. These are people over the years that have contributed to that work, and this is my team at the Memory Center and some funding. So happy to answer any questions.